For this lab, we're going to be separating a mixture using a technique called chromatography. Chromatography relies on the difference of certain substances adhering to some kind of a stationary phase in a different way. And we're going to be separating the pigments that are in leaves because leaves have two different forms of chlorophyll, chlorophyll A, chlorophyll B, and also have beta-carotenes and xanthophylls, which produce different colors, especially around this time of year when we start to see the leaves changing colors. We're going to use 70% isopropyl rubbing alcohol with water as our solvent. That'll be our mobile phase. And then, as you can see, I cut up some leaves and place the leaves into the rubbing alcohol to increase the surface area to help extract the pigments and make a homogeneous mixture of those dyes just a little bit easier. You can also see over here I have my hot plates. My hot plates I have set to high right now because I want that water to be close to boiling, but I'm actually going to turn the heat down to the medium setting here. And the reason why we're using a hot plate and not a Bunsen burner is because we want this heating to occur very slowly to slowly extract those pigments into our mobile phase so we can use our stationary phase to begin to separate them. Okay, so I think our water is hot enough, and I'm going to place the beaker in the hot water bath for the time being. We're using the hot water bath to indirectly heat the rubbing alcohol. Obviously, rubbing alcohol is flammable, and we also want this process to occur very slowly. So I'm going to let this sit in here for about 10-20 minutes. We'll see how much of that pigment we actually are able to extract, and then we'll start to set up our stationary phase in a moment here. As you can see here, this is our experimental setup with the piece of filter paper that has been cut to fit inside of the beaker. I actually drained the liquid from the leaves just so we have a nice even surface with which the dyes can begin to move up the paper, and you can start to see inside the paper that it's starting to move up the stationary phase. The mobile phase is the rubbing alcohol that now has the pigments in it. You can see that I used a pencil just to suspend the piece of filter paper so that it's just barely touching the liquid. And you can also see that I drew a line in pencil at the very bottom of that filter paper in order to have a reference point to see how far up the piece of paper the dyes traveled. We're going to give this a little while to separate, and then after it's done separating, we're going to take a picture of the actual dyes to measure how far up the piece of paper they traveled relative to one another.